Good afternoon, everybody. My name is Zoltán Molnár. I'm professor of developmental neurosciences here in Oxford. And welcome to this Neurotech event, which is basically a quick introduction to digital monitoring in health and disease. So I shall make an attempt to share my screen. Give you a very brief introduction to this event. So I'm extremely grateful to Professor Kristalina Antonides and also Dr. Andre Marcus-Smith and also Ms. Marissa Müller for joining us today. This is a warm-up event, and hopefully many more will follow after this event. But this is giving you an idea of the kind of activities we are planning as part of NeurotechEU. So I am in charge of work package 6 for NeurotechEU, and this is very much part of this public understanding part of Neurotech. So digital monitoring in health and disease is extremely popular and getting more and more popular every day. If you think about it, we measure our activities when we do exercise. So during the weekend, I used 622 calories on this stepping machine. But when I went to my zone, then the calories were 1,382, which I was very pleased with because obviously there is no standardization. And I was even more pleased to see that this put me in the lead because I'm competing with 15 other, actually here 9 challengers, mostly rowers. So we use this digital technology all the time. When you ski, or when you do some kayaking, you're running around or walking, you use your Strava, or you even monitor your sleep from the noises you emit during sleep. So here you can see that this was a particularly good day, 97% sleep quality, with whatever that means. So we use these technologies all the time. And this is becoming part of our life now. We can measure blood pressure, weight, we can measure our temperature, heart functions, oxygen saturation, and we have all sorts of variable monitors which we use and take advantage now. But these technologies will become extremely useful when we want to monitor conditions of patients, and Kristalina is going to talk about this, how to monitor a Parkinson patient using these devices, how to set up medication, how to keep an eye on them. These technologies are now part of our life. And also elite athletes like Marissa is also very interested in knowing her performance, whether she's improving. So these digital technologies are also extremely important for elite athletes. And if you look at these monitoring devices, they become part of a life now. So you can measure your heart rate, oxygen saturation, breathing, muscle electrical activities, your stress, your skin conduction, cognitive functions, movement patterns, and also the sleep. So you have all sorts of smart shoes, smart socks, smart pants, smart glasses, which you can use to monitor your activities, and then you can record it. Now, together with these devices, we also now have specialized medical devices and wearable equipment. And also the apps are becoming better and better to monitor these activities. And also, there are all sorts of ethical questions surrounding this. Who should be able to see, what I did on the weekend, in my Strava or my zone? Is it the friends, family, everybody, doctors, nurses? Because this will have an impact on how I'm viewed. And so these ethical issues come up immediately. So today we are going to have 3 talks. We are going to start with Professor Kristalina Antonides, who is from the Nuffield Department of Clinical Neurosciences. Kristalina did a PhD at Cambridge, and then came to Oxford and set up her own laboratory. And her strength is to look at this distant monitoring in Parkinson's disease. And her talk will be giving an introduction to this topic. Then Andre Marcus Smith is going to talk about scaling up neuroscience and psychiatry, a perspective from tech-informed by a career in academia. So Andre did an MSc in, did first a psychology degree, then did an MSc in neuroscience here in Oxford, then a PhD at Oxford, then became a group leader at the Wellcome Center in London, and now he has his own company where he is a chief technical officer at Neurotechnology. And he's going to talk about some of his approaches to this. And finally, we will have Marissa Müller, who is a MD PhD student accepted to Harvard Medical School, but right now she is a Rhodes Scholar at Oxford, and she's going to talk about algorithm choice influences accelerometer measurements and clinical implications. So as I mentioned, these events will give you a taster of what will come as part of NeurotechEU, mostly linked to Work Package 6. So I'm very grateful to Kristalina, who will give the first talk.
Thank you, Zoltan, and thank you for the great introduction, telling us a little bit more about digital technology. Let me just, Nick, can I share my screen, please?
You should have no problem sharing.
Okay, there you go. Can you see my screen now?
Perfect.
Yep. Okay, great. Thank you. It is a great pleasure to be here today to give you this short talk on digital phenotyping in Parkinson's disease. I'm a clinical neuroscientist, and my area of expertise really is the development of biomarkers in neurodegenerative conditions, and my special interest and training is in movement disorders and Parkinson's disease. And today I'd like to tell you a bit more about the work we've been doing on quantifying Parkinson's, really, because this is very much what digital phenotyping is in neurology and clinical neurosciences. And by quantifying Parkinson's, I mean using digital technology to help us record things like eye movements, oculomotor function, and gait abnormalities. And of course, with a very short sort of taster talk, as Zoltan said, is impossible to cover everything, but this is just to give you an idea of some of the work that we and others around the globe have been doing and the potential that there is in digital technology and digital phenotyping. Just to set the scene, particular for those of you in the audience that might not be familiar with the neurodegenerative condition of Parkinson's, Parkinson's, along with Alzheimer's, is one of the most common type of neurodegenerative disorders we see. And with the number of aging population increasing, Parkinson's presents more often and in some instances at younger ages. And Parkinson's is classically defined by a pathology that targets the dopaminergic cells in the nigra, in the substantia nigra, as you can see here. And these cells acquire an alpha-synuclein Lewy body pathology, which I hope you can see my cursor down here. You can see in this figure. And whilst this is a very useful approximation of Parkinson's, and we know that if we give dopamine drugs, for instance, which target that particular network, some patients may do very well and have a really good quality of life, it is not the whole story. And just very briefly, to take you back to 1817, when James Parkinson, who was a British physician, firstly described in his beautiful essay, which we can find here in Oxford in the Bodleian Library, he called an essay of the shaking palsy. And in fact, it wasn't until the 19th century that another neurologist, Jean-Martin Charcot, began calling it as Parkinson's disease. It took about 100 years for doctors to realize that the patients were actually losing brain cells, and it wasn't until the 1960s that it was actually discovered that levels of the brain chemical dopamine were much lower in these individuals that had Parkinson's disease, these patients, as opposed to healthy controls. And what I mean by healthy controls is very much the people who do not have a neurological disorder or a psychiatric disorder for that matter. And of course, if you're interested a little bit how it was described over the last 200 years or a bit more, Sir William Richard Gowers, who was a neurologist, a researcher, and an artist, drew this illustration in 1886 as part of his documentation of what he thought Parkinson's looked like. And in fact, up until recently, we've all considered Parkinson's to be an elderly disease, something that it doesn't present itself earlier on, as you can see from this picture here, with this posture sort of leaning forward. You can see this person probably has rigidity, the upper limbs are looking quite stiff, and also this Parkinsonian face, this face mask, as we call in neurology. And what's nice is that more recently, Dr. Johnny Akerson, a lovely consultant, sort of emergency physician here in the UK, has redrawn a lovely picture to show how we now understand, or at least start to think really, the sort of both sides of Parkinson's, both the motoric and the non-motoric. And by that, I mean, not only things like tremor and problems with movement and so on, but all the other things that the Parkinson's patients report to us, problems with being apathetic, having depression, cognitive problems that perhaps we might have overlooked over the years, but more recently, lots of us have started looking into this, which has been tremendous to have an updated picture as you see here. So there's no cure for Parkinson's, unfortunately, but there are treatments that help improve some of the symptoms and that's the quality of life of the patients. On the one hand, I show you here, we have various medications and various treatments. The most popular one, including levodopa, that can have an effect, a therapeutic window time frame, while on the other hand, we have a surgical technique called deep brain stimulation, which involves electrical stimulation of deep brain areas that can relieve symptoms. And because a picture is 1,000 words, in this case, I'm going to show you a very quick video of what a patient looks like when you switch on and off their stimulator. So the electrical stimulation that's provided in those parts of the basal ganglia, this particular individual is in somewhere called the subthalamic nucleus, which is part of the basal ganglia circuit. So let's have a look.
John Radcliffe Hospital in Oxford, where the deep brain stimulating device thing was put in. You're going to switch it off?
Certainly can, yeah.
Okay, over to you, John.
Quite a violent tremor, as you can see. I think that's enough. We'll put it back on. Stretch your arm out, and you can see almost straight away.
Oh, that is absolutely astonishing. It's fantastic.
So I hope you can see how quickly that tremor can be eliminated, if you like, at least for a short period of time by the electrical stimulation switched on and off. But unfortunately, that's not the case for everybody. And the surgical technique, including also the medication treatments that are currently available, might not be having the effects that you see from this individual here. So for the DBS, for the deep brain stimulation I've just shown you there are various targets. And just very briefly, the most popular ones are the subthalamic nucleus and the globus pallidus internus for Parkinson's. And if you're looking at primarily tremor, you can target the VIM, and an older target was the pendicular nucleus, PPN, that was used various aspects of Parkinson's. But STN and GPI are the two nuclei of choice at the moment, which seem to be working well if the surgery is successful and if the patient has sort of acceptably sort of gone through the inclusion criteria to be included in the surgical treatment. And some earlier trying to contact in this patient population and compare it with individuals who don't have a neurological disorder. And the rationale for doing this, especially the motor function on the quantification of eye movements, is simply that many of the basal ganglia areas that are involved in saccadic generation are also closely related to some of these targets I've shown you that we put deep electrodes. And particularly, I show you here the subthalamic nucleus, sort of sending excitatory input into the substantia nigra pars reticulata, and the supracolliculus down here that receives tonic inhibition from substantia nigra pars reticulata, and in turn is inhibited by the caudate nucleus. In simple terms, for those of you in the audience that might not follow the basal ganglia circuit, this is very much closely and intimately related. So anything that you're doing in this area here will be having either a direct or an indirect effect with how you're moving your eyes, how you're responding to things. And if there are any abnormalities, obviously, we should be able to see what those are. So now I'd like to tell you very quickly about our study called OXQUIP and how using digital technology has been incredibly helpful to advance our knowledge to date. So there have been many biomarker studies in the area of neurodegenerative disorders, and OXQUIP that you see here is short for Oxford Quantification in Parkinsonism. It's a longitudinal study that follows patients both with Parkinson's, but also an atypical type of Parkinson's called progressive supranuclear palsy. I will be calling it PSP. We are following patients very frequently with assessments every 3 months, with identifying a signal of progression at a much shorter time scale in order to be able to rapidly screen newly developed drugs. This is really important because only a small minority of drugs that have already been developed actually make it successfully through trials to the market. And developing and trialing such drugs consumes a vast amount of resources. The aim, therefore, for this is to kill off research into doomed drugs quicker and divert, if possible, both time and money to the next target, if there is one really, and if not, to what's up next in the horizon. In OXQUIP, we're neurophysiological and cognitive signals to find physiological correlates of time scale of 3 to 6 months. We're also focused on ensuring that the baseline diagnosis is as accurate as possible because it obviously is critical to get the right patients into the correct trial to begin with. And just briefly, let me show you the type of equipment and the type of digital technology we've been using that's been tremendously important to help us identify some of these signals. So technological advances have enabled us to measure some neurophysiological signals very easily. In OXQUIP, we measure rapid conjugate saccadic eye movements using portable devices such as the saccadometer. So a saccade is the most common type of eye movement we all make. You're all making it at the moment. You're looking at your screen, you're looking at me, you're looking at the slides, you're looking at all the things I have on the slide. So this is how quickly you shift your gaze from point to point to take in all the information. We have a wealth of information on this from people who don't have neurological disorders over the years, both physiologically in humans and in macaques. So we know very well how eye movements should be behaving. So anything that is abnormal is usually due to a disease sort of symptoms, if you like. So what you see here on the left-hand side is what I've just referred to as the oculometer that we use to measure and quantify your eye movements as quickly as possible. So what you see is a portable oculometer, so you don't really need to come to the lab or to a specific clinic. We can take this to our clinics. And it's an elastic band that fits around your head. There are 3 LEDs, 3 lasers. They're protected on any screen or surface or even a wall in this case in the clinic. And this device has 2 infrared cameras. And the infrared cameras here are measuring exactly when your eye is moving to either direction. Similarly, we are measuring hand tapping that you see here very accurately using touch sensor devices and trying to digitize clinical rating scales such as the UPDRS. This is the short version of something very fancy called Unified Parkinson's Disease Rating Scale, but really this is a clinical questionnaire. So when you see a patient in clinic, you ask them to touch their nose, touch your finger, open and close their finger as fast as possible. So this accelerometer here that you see this patient wearing on their finger here is precisely measuring that movement to essentially add to the clinical confidence when you're seeing patients like this. Also, we're measuring cognition that you can see here. and is measured in this case using computerized tablets as opposed to the pen and paper that we usually see in neurology. And of course, when you have a tablet, you can see things like reaction times, you get spreadsheets behind this. So the data that you get would have been impossible if I just handed a pen and paper to the patient, which can be useful if you don't have a computerized version. But again, this is extremely useful in what we're doing. Last but not least, I hope you can see my cursor down here on this wearable sensor array. We measure gait using a body-worn array of inertial sensors containing both accelerometers and gyroscopes. And just to show you what these look like in action, this is a lovely patient who has accepted to show what it looks like when we bring them to clinic. So this is myself in one of the neuroscience outpatients here in Oxford, taking this portable accelerometer and measuring really some of the eye movements very easily and very quickly by projecting this red light on the, we didn't have a screen there, it's just the wall of the side of the clinic. And of course, I choose a clinic room that doesn't have a lot of windows, so we can minimize luminescence. So it's very easily done and very quickly, and is very easy for the patient as well. They don't feel that they're sort of clamped in, if you like, if they were to come to the lab to some of our older devices and more clunky devices. Moving on to the sensors, this is what the sensors look. You placed here, and I'll talk to you in just a moment in a couple of more details. Similarly, we have a sensor on the chest, one on the lumbar, but all I want you to take away from this is the wealth of information and really the big data sets that are coming out of this. That again, I think Zoltan alluded at the introduction. It's interesting, but it does elicit a number of different questions. Who looks at this data? How do we treat this data? And how to take this area forward, making sure that we analyze the data as accurately but as fairly really for the patients as possible. Very quickly, a couple of the results from both the eye movement first and the gait. So I show you what this data really provides in terms of the translational aspect of our work. This particular study came out from OxQuip, and it's very much looking to characterize the effects of anti-Parkinsonian medication on eye movements. And here you will see two words, prosaccades and antisaccades. So prosaccades is when you move your eyes to really do a reflexive eye movement, which is what you're all making at the moment, looking at the screen and listening to this talk. We know a lot about it, and we know what is within normal range and what is not. Similarly, an antisaccade it's a more complicated task, really. It's something that you have to move your eyes to the opposite direction from where the light is, and it involves a higher order function, really, a cognitive function, to be able to stop yourself looking at something and do something in the opposite direction. And just in one slide, what we found is that this reflexive movement, this prosaccadic latency, we call it, is significantly affected when we give patients dopaminergic medication, which in fact may complicate using eye movements, for instance, with these reflexive eye movements as a biomarker in drug trials. But what we found, and was really interesting in this paper, is that the antisaccadic latency, so this idea of moving your eyes to the opposite direction, is particularly interesting in this regard, because it shows a large disease effect, which you can see here, with no medication effect. So what this is showing is that had we not had digital technology to measure eye movements and measure really the oculomotor function of our patients and compare it to those people that are referred to as healthy controls, in this case they were the spouses, we wouldn't have found that using an antisaccadic trial would help us to the extent that we could potentially use this as a biomarker in clinical trials. Very happy to share the papers if anybody's interested. And my last point in this short talk is moving to the more digital technology in terms of sensors and sensors around the body. So this study, again, part of the OxQuip trial, it is important that we are able to do, to accurately and as early as possible, really, identify future disease-modifying therapies both for Parkinson's and PSP, these disorders that look like and mimic Parkinson's, at least at early stages of the disorder. So analysis of gait and related tasks is one possible means of discrimination, and this was the purpose of this study. So this was published in Gait and Posture, and what we investigated here was a wearable sensor array coupled with machine learning approaches as a means of disease classification. And let me just show you what we did. We analyzed gait, so how people walk and the behavior of gait really, in a group of people that had been clinically diagnosed with Parkinson's and also clinically diagnosed with PSP. Again, this is a bit difficult because especially for PSP, you cannot be 100% sure unless you have post-mortem material, unfortunately, but we did it on the basis of clinical diagnosis, which is the best we have at this stage. So we used a body area network of wearable accelerometers. And a bit more detail on what we actually used. And I'm using this GIF here to show you a little bit what it looks like. All participants completed a 2-minute walk. You can see one of my wonderful DPhil students here, Lily, wearing the 6 sensors. So they did a 2-minute walk on a straight line. Next, we measure the sway. Participants were asked to stand upright and as still as possible for 30 seconds with their eyes closed on a firm surface. And you can possibly see a template that was placed on the floor to ensure that all participants' feet were the same distance apart. And then this last one here is a timed up and go that was repeated 3 times, each repetition until the participant standing, as Lily's doing here, walking till the end, turning around, and then coming back and sitting on the chair. I should say that for this particular study, we used 6 sensors. So you can probably see Lily wearing something like a harness on her chest. There is one on the lumbar, on the lower back. There are 2 watches, one on each arm. You probably can see this when Lily turns around. And there's also 2 sensors, one on each feet, which you can't see very well because we have black shoes against black sensors here. What we found was that we had 17 distinct gait parameters that separated PSP, this is a typical Parkinsonism disorder, from the other PD and healthy controls. And what was interesting is that when you use different sensors around the body, like the 6 ones I've just shown you, we could have an accuracy of up to 90%, which at that stage was better than any other test we or colleagues have done, including imaging, blood, and CSF tests. And just very quickly, just to show you what this data looks like, because a lot of people usually ask me, what do these data look like when you're measuring it? This is an example of some of the data available from the gait and sway. You can see the parameters here derived from tasks overlaid onto the normal ranges shown in gray, you can see it here, which make the features that are abnormal very clear. So you can see all these left and right for the lower limb, left and right for the upper limb, being outside the normal range. Of course, you can also see these wiggly lines. This is the sway. We all have physiological sway, but this is very much abnormal. It's sagittal versus coronal here, where we're moving either forward, backward, or side to side, which again, we wouldn't be able to have data of this sort to compare and contrast. And of course, remember every 3 months that we see these patients. The timed up and go, here as you can see the parameters derived from getting up from the chair. And again, this is the time that it takes to turn, for instance, the time that it takes to stand to still, sit to stand, and so on. So again, we wouldn't have had this opportunity to know this exact data. And I'm going to finish off with just saying a little bit about the importance in trials and how important technology it is for us. So for the study of PSP, we need better ways of identifying the PSP patients from the wider Parkinsonian population, really. And of course, for Parkinson's trials, we need to remove not only people with PSP, but other atypical Parkinson's from the study population. We get a lot of information. What's the minimum number of sensors? And we can discuss that later with the colleagues that are giving talks later and with everybody who is attending here, how you can determine what is the number of the sensors that one can use, because there's an international discussion going on this particular point. And for best diagnostic performance, you do need a larger number of sensors, but it can be reduced depending what your question is and what you're trying to achieve. And of course, for monitoring, once diagnosis is secure, you could sort of reduce the number. And this is my last slide here, just to summarize. So technology can help us quantify eye movements and gait abnormalities with great accuracy. Digital phenotyping can be useful for personalized and continuous monitoring. It may help to improve decision-making, making it in the measurement-based care. For digital phenotyping to drive benefits in neurology and of course in other clinical areas, serious considerations must be given to the practicalities of future clinical application. Digital phenotyping must be easy to use, to my mind, and really it must fit in the established quality and safety in the discipline. And of course, it must be cost effective, which is what everybody is sort of striving on at the moment. We certainly need, again, to my mind, global leadership, collaborative efforts to make this work effective. And we definitely have a lot of work ahead of us. But I do think there has been significant advances in the last few years. So do watch this space. Thank you very much for listening. This is work done by the wonderful Foxquip team. We do have a lot of fun working together, and I'm very happy to answer any questions at the end of this session. Many thanks for your attention.
Thank you very much, Kristalina, for this wonderful talk. I hope you will submit questions to the chat function, and we will have a general discussion at the end of the session. But now we move to Dr. Andre Marcus-Smith's presentation.
Okay, thanks very much, Zoltan. And thanks to the audience for making the time to be here and for the invitation to speak as well. Zoltan's done a pretty good job introducing me. And I would only add to that currently, unfortunately today, I'm not going to be able to talk about what we're actually currently building at my company. I'm more than happy to do that at a later point when the work isn't confidential anymore. And so today I thought what could be useful is if I provided, kind of our own perspective on what small tech companies such as ours can bring to neuroscience and psychiatry, and how we can have collaborative dialogues going forward. And speaking about collaboration and dialogue, what better way to start that than with provocation? And to the psychiatrists in the audience, what I would like to ask them is, to consider if there's another specialty in medicine where we accept the status quo of patients having to undergo weekly treatment, sometimes for years, having nothing better than a guarantee, having no guarantee for cure or remission, rather, and only maybe a modest attempt at symptom management for the rest of their lives. And to the neuroscientists, something that I sat through many times during my career as a researcher, which is how many times have you sat down and you've had your aunt or a friend or your granny ask you, why is it that she gets so anxious when X and Y happens, or what goes on in their brains when they're craving popcorn, or whatever other type of question that immediately you feel like I should be able to give a straight answer to this, but it turns out to be actually quite complex here. And you end up couching all of your answers in, oh, it's complicated and X and Y and so on. And you just feel this sense of inadequacy is what I always felt in these situations. And what I would like to start out exploring here is first, why is this? Second, why are we okay with this status of affairs? And finally, whether it has to be this way, whether it's something that we can do as professionals in the field to move this a little bit further. Naturally, on the why side of the question, things are complex. It's very easy to make provocations like this and alienate 97% of your audience. But as we all know here, there's a huge amount of variability in brain micro and macro architecture, functional architecture, and patterns of activity, both between individuals and the same individual at different points in time. Even a week apart, you can get brain scans that are completely different to the same task in the same participant. And as all the experimentalists in the room know, nervous system recordings are, well, pretty difficult to do, regardless of the technique. And for this reason, we don't have the dentist weekly, not weekly, unfortunately, yearly checkup equivalent in neurology or psychiatry or clinical psychology. People don't go to these specialties until they have a problem. So we don't have any of these intermediate data points from the beginning of a phenomena to the occurrence of the phenomena, before that, and all the way through to the remission or treatment ending and so on. And what we also know is that doing recordings of the nervous system, central, peripheral, what have you, is both cost and labor intensive. And it's inherently quite difficult to scale, even in animals where we don't have a lot of the constraints that we have in human beings. And these two factors together, I think, have contributed to getting us stuck in this small data regime. So all this revolution that's been happening in terms of how we do data science, how we do machine learning, some of it has filtered into neuroscience and mental health, but a lot of it we're still not able to deploy because we don't have the huge types of data sets that are required by these approaches to function well. I think another very important factor that all too often we overlook is that a lot of the data that we're collecting in the laboratory, be it from animals or from humans, is not really reflective of the way that the phenomena we study tend to happen in real life. And here I would point, for example, in the case of systems neuroscience, the way that we study decision-making and problem solving in animal models, for example, is absolutely nothing to do with the way that those factors actually play into the animal's ecology. And this is also true when we study other more human forms of processing as I'll comment later on in this talk. And finally, for these and many other reasons, biomarkers, especially for mental health, in neurology, the situation is different, but in psychiatry particularly, have been very difficult to find. And this is a really, really huge stumbling block. I mean, the analogy that I always draw here is, where would diabetes treatments be if we couldn't quantify blood glucose accurately? And moving on from a somewhat pessimistic and gloomy picture, I'd rather focus on what we can do about this and how can we improve this state of affairs. And I think the first factor on which we can get a lot of headway, and a lot of things are happening right now even, is on hardware. My own perspective on this and the perspective of our company is that the key priority, the key focus should actually be to build hardware that can enable us to get research grade data, but outside the lab, in ecological conditions, in the places where people actually live and carry out their daily activities. And what we think here is that you can really look at hardware as a platform rather than a tool. And it's a platform that can enable for you, if you're able to pull this off, on one hand, collection of data at an unprecedented scale, on the second hand, improving lives of patients by having them do those more continuous type of checkups, not by having to go to the hospital, but from the comfort of their daily lives. As Kristalina was showing, this is the kind of thing that can even uncover biomarkers and differences between patient populations that would have completely been missed had you not been able to do this type of monitoring. And lastly, this can help us answer our grannies and our uncles' questions as well when they ask you, oh, what goes on in my brain when I go and do choir singing, or when I'm getting really upset at talking to that annoying upstairs neighbor or whatever. These are things that matter, actually, if we want people to be interested, if we want people to have a closer relationship with their brains and minds than they currently do. The second point that I would make on hardware is that I think, especially from the side of the tech industry, it's very easy to fetishize technology and sensing capabilities. And any neuroscientist, especially on the experimental and system side, will know that new modalities for doing brain recordings, especially non-invasive, are few and far between. In really key groundbreaking innovations that enable us to measure the brain in a completely different way from before are things that happen every 10, 15, 20 years. And they will continue happening, and that is great. And we will record more neurons with increasing scales of resolution. But as a neuroscientist, I also know that we are not at the point where this is simply an engineering or resolution problem. In the limit, as someone coming from the systems field and used to working at the cellular and subcellular scale, if you told me, okay, Andre, you can record with 1,000 neuropixel probes and get 1 million neurons from this awake behaving mouse doing ecological behaviors, the first thing I would do would be dimensionality reduction so that I could actually understand those 1 million neurons as if they were 100 or 1,000. So we still need to do a lot of scientific work, actually, before we simply start treating everything as a resolution or hardware problem. And there's a second point that I would bring up here, which reflects a bit my journey as someone coming from a fundamental science background into a technology more applied setting, which is that my feeling is that in systems neuroscience and in the research neurotech environment, we've developed absolutely spectacular tools for interrogating the nervous systems of rodents, namely mouse models, with amazing levels of precision. But my concern is that there is a gap in translation. And a lot of these technologies that we've developed, which are extremely insanely powerful in rodents, are very unlikely to ever see application in human beings. And whilst it's clear that we need to develop technologies for the sake of fundamental science and making discoveries in rodent cell types, for example, I would like to see that balance tilt a little bit further into tools that we can use across species and actually eventually bring over to humans as things that can actually be impactful. And finally, on the sensor side, I would say that rather than spending a lot of time developing new types of sensing and high resolution and so on, I think there is already a more pressing need that we can devote our attention to, which is simply the fact that we need data. We need a lot more data than we currently have. If you look at the, a large human study in neuroscience is something between 1 and 10,000 participants. 10,000 participants is an insane number in the context of human research. And then, but you imagine that, and if you think of the number 10,000, and you think of that in the context of a wearable, for example, if a company sold 10,000 wearables, that company would be out of business, right? So when you start thinking in device numbers and data sets that you could potentially reach with a hypothetical device for doing brain measurements on the go, you're really starting to take this number and scale it by a factor of 1,000. And if you look at the types of data sets and the sizes of data sets rather, that modern machine learning has had a huge amount of success with, for example, on the computer vision side, we're really in the realm of millions, tens of millions, hundreds of millions of samples. So really we have to start trying to take neuroscience into that regime, not the little data regime. But again, as with everything concerned and even remotely touching upon neuroscience, everything's really complicated. We don't just need more data, we need data of different kind. And I think our model of epistemology in neuroscience has been too closely modeled on the physics side of the scientific method, I would say, in the sense of running very controlled, highly structured, linear interaction type of experiments. And that's really good, and that's brought us a very far way, but I think the balance is still too far away from experiments where you forgo some of that control in order to achieve higher degrees of ecological validity. And the reason I think this is fundamental is not just a nice point to have on the final line of your grant application, is how do we know what we know about the human brain and emotions, for example. We put people in an fMRI scanner or with an EEG headset or something like that, and we show them movies of sad things or happy things or outrageous things. And this is supposed to model the emotions, the sort of turbulence that goes on inside us and takes over us when we feel these emotions in real world settings. And as we all here know, and as the researchers doing this type of research to the best of their knowledge know, they're worlds apart. But currently that's the best that we can do, so that's what we do. But we need to move on from that into ecology. And lastly, on the point of data, I would also say that the fact that we've been stuck in the small data regime has meant that we've needed to do a huge amount of feature engineering in neuroscience. So typically we're looking at patterns of activation in bold. We're looking at event-related potentials. We're looking at powers in different spectral bands. And all of these features are powerful. They're interesting. Importantly, they're mechanistic or at least theory-driven. But if there's one thing that we can learn from the field of computer vision is that humans suck at feature engineering. There's no guarantee that these are the best features that we can get from an EEG or an fNIRS recording or an fMRI scan either. But in order to go into this data-driven regime and finding out, okay, what are features that are even more predictive than the features we've been using, we need to get into a different regime of data collection. Now, the way forward here, I really think has to do with collaboration. And it's a really interesting point in time in the sector of neurotechnology, because we've already seen a few mishaps in communication between the world of brain researchers and the world of technologists, names starting with E and ending in Elon Musk, for example, where clearly there was a shock between two worlds. And clearly there were some things that were lost in translation. And I think the thing that is really good about tech is that tech builds well. tech scales, tech can move fast, efficiently, it can cut through red tape, it can just get things done. And that's a huge asset. It's a huge power to be able to do that. It's also ambitious, which is fantastic. But it also has a certain proneness to falling into traps in terms of short-term profits and gains. Tech is also good at very quickly, efficiently, and scalably building bullshit and losing scientific rigor. And you can see this by the amount of devices currently on this very market, which offer, make promises in terms of gains that you can achieve on your focus, on your memory, etc., in ways that no serious neuroscientist could possibly countenance, whatever work. On the flip side, academia and neurology and psychiatry are evidently where the top minds and the cutting edge thinking in thinking about the brain and mental health live. But often people in these areas are either uninterested in taking on real world problems, or if they're interested, they don't have the tools, they don't have the resources to actually build things at scale and have a big impact. It's not what a university is necessarily designed for, pretty much. And I think what is really interesting about this emerging sector is that a neurotech company will live and die by the quality of the algorithms that it puts out. And drilling that point down even further, and I think this is really cool, really amazing, companies will survive or die in this field depending on how well they can decode activities of the brain into behaviors or states of mind. And that's a scientific question. And it's a scientific question that's going to have a direct impact on a business and on a human and societal level. And that's amazing. That means, we as neuroscientists can finally come in and have an incredibly meaningful and strong impact on the day-to-day runnings of society. And personally, again, this is my personal take and kind of my company's position, but I think that what we need to try and achieve here in this sector is combining really this key strength of tech, which is the infrastructural, technical, and financial resources that we have with intellectual resources that live within the halls of academia. And in more practical ways, I think what this means is that we need better frameworks for collaboration. I don't think grants are necessarily the best way forward. If you're a company and you're needing to spend time putting a grant application forward, and then they tell you, oh yeah, there might be like a 20% acceptance rate. If we're lucky, like, I'm not going to spend, time and money for a 20% flip of the dice. We also probably need faster project cycles. So there is a lot of interest from the industry side, I think, in commissioning and sponsoring research, but the 3 or 4 year timescales associated with a PhD or a postdoc are extremely long in terms of tech roadmaps. So I think thinking about timescales on the range of 3 to 9 months and thinking of placements, secondments, the UK has these knowledge transfer fellowship, which I think are a really, really good innovation, is probably the better way to go. And then from the universities, of course, there's two sides to this question every time, but we also need policies which are realistic and not predatory in terms of IP and equity taking. And I don't know if this is the case necessarily in the UK, but at least in a lot of EU countries, universities often take a very large stake, sometimes up to 30 or 40% of a spin-off startup's equity. And this can effectively just make a business unviable from day one going forward. And lastly, another thing that I think is starting to happen a bit more now, but I think we need to do it even more, is exposing to our students, especially our graduate students, this scientist, entrepreneur, and career path. It's exciting. You're still doing science. You're not doing it at university. You're doing it in initially a small company and hopefully a bigger one then. But it's been striking to me how many qualities that were trained and drilled into me as a graduate student have actually turned out to be super applicable in this new world. And lastly, I think we need to think carefully about ethics, but we need to think about ethics also in a real politic way. So we need to have in mind the goals and outcomes that we can achieve in this field, more than strict, pure ideological considerations. And here we need to talk about admissible uses of data. We need to talk about privacy and security of how these types of nervous system data are kept. But we also need to bear in mind that if we introduce regulations which are too draconian, we're severely going to be limiting what we can do and how many people we can help with that data. So we have to strike a good balance here, somewhere in the middle. From the company side, I think the impact that continuous remote monitoring of nervous system activity can have is so huge that it needs to be a founding principle that accessibility and affordability of these things is built into business models. This can't just be the privilege of the rich and wealthy to do going forward. And from the academic side, I would say in these collaborations, the focus has to be solving problems. We need to be helping someone do something better in their lives, feel better in some way, have a better treatment outcome. And papers will come later, that's fine, but let's put that second stage. I will, if I want people to get excited that, I can collect your 10,000 hour data set on sleep from 100 participants or something like that. I want the first question to be great. When can I get my hands on it? Not how are we going to publish this? And since I did my talk kind of back to front, I thought it would also be useful to, as I was alluding to earlier, this path from science into entrepreneurship, at least in Europe and the UK, is still not necessarily a very common one. So all I can talk about is how I got into this, whether that was a good route into it or not. I don't know, it's worked out for me. But as Zoltan said, I came from initially psychology, especially clinical neuropsychology background in Portugal. I came to Oxford for the Wellcome Trust PhD program, and my research was with Zoltan and Simon Butt, focused very much on developmental neuroscience and circuit mapping. I stayed on this field for my first postdoc and then transitioned more into systems neuroscience research. So this was the point where I started having a more direct contact with developing and building technologies for recording from the brain. I was involved in a few projects with Adam Camp at the Sainsbury Wellcome Center, including the NeuroPixels project. And to me, this was one of the pivotal moments that got me thinking about building things. So the seed was set at that stage. And I got a Henry Wellcome Fellowship after that. But this was very much, all of this work was fundamental science. And this fellowship was also fundamental science problem. But this was the point where I started thinking, I love the work that I'm doing. It's fascinating. I'm surrounded by incredibly interesting people that I learn from every day. But I kept feeling like something was missing. I kept feeling like I wanted a new challenge, that I wanted my work to start being less arcane in a way, and to start being something that was more applicable, more related to real life. So I sought an opportunity initially at another person's non-invasive BCI startup. And that was a great experience. I learned a lot working at this company in London. I was one of its earliest employees as well. And during that period, and this period was very useful, and I could not emphasize enough the value of having a first experience outside of academia, because it's a completely different way of working. But during this period, I not only learned a lot about what a startup is, what it means to work at one, the challenges, but also the good things that you face and what you need to change as a scientist in order to fit in. But I also started forming a different perspective on the way I thought the neurotech field could play out. And it was at that point, after a period of reflection, which I think is always a fundamental thing to have in life, that's when I decided to, after forming up my own perspective, start my own company. I'd be very happy to answer any questions, either now or later in the discussion time. And that's my email, if anyone is interested in getting in touch. But thank you all for listening.
Thank you very much, Andre, for this very interesting journey, not only in science, but also career, which is very good to hear. And now we move straight to Marissa's talk, Marissa Müller. But please keep putting your questions into the chat function so we can have a general discussion after Marissa's talk.
Thanks so much, Zoltan. So before coming to Oxford, I competed internationally in athletics while studying engineering. So I started working with accelerometers in Dr. Freilaw's neuromuscular biomechanics lab, University of Iowa, which I thought was great. I used accelerometers every day when I was training for sport, as we got a glimpse of with Zoltan at the beginning. So the devices sense motion and they turn raw data signals. So this is what they look like, these random lines, into numbers that we care about, taking a dive into what's actually happening, the number of calories you burn, the steps you take, or the time that you spend in moderate to vigorous physical activity. So these should be objective measures of activity, which are important as we've seen across recreational performance clinical applications. It's the clinical relevance that we ultimately care about, like what Kristalina was speaking of earlier. But to begin this talk, I want to talk about the actual devices and a little bit of the math that happens behind the scenes. So finding better ways to measure physical activity using accelerometers, as we've seen, it's become a very popular research topic. It's a multi-billion-dollar industry. And the accelerometers require lots of different equations and data filters to interpret the activity. This is a very, very small sampling of some of the math. It's complicated, but for today, it's really not important to know any of the fine details. But what is important is to know that previous studies looked at different sets of these equations, which are supposed to calculate the same outcomes. For example, the calories you burn. And it appeared that different algorithms return different outputs when applied to the exact same data set. So this isn't between your wristwatch and Zoltan's treadmill or step count. This is with the exact same device, just using different equations, and this isn't supposed to happen. So I really started paying attention to this idea of algorithmic variability when I was analyzing raw accelerometer data from a study that looked at how engaging in physical activity might influence chronic pain in patients with fibromyalgia. And the results just weren't making sense, and they changed a lot depending on which equations I used. So again, looking at the raw data and the math, I hypothesized that these differences had to be greater than what had been realized in the literature. And I wanted to answer the question, how exactly do the accelerometer algorithms impact the resulting measurements? So we conducted a study where participants wore an accelerometer for 7 days and they completed self-report activity logs. So I analyzed and compared data using different combinations of a filter called the low frequency extension, wrist correction, and multiple popular equations. So we found that different algorithms produce bigger differences than we thought. So this graph highlights some of those differences in the number of calories that are reported as being burned. So for Zoltan, it was like 630 versus 13 something. Here, for the exact same data sets, we have 1,000 versus almost over 3,500, and that's a massive difference. So this risk correction was not applied consistently. It really decreased estimates, and in these two cases here, it had no effect. And then on the opposite end, the low frequency extension increased steps by quite a lot, but didn't do anything else. And this graph shows the same information, highlighting the effect of steps. And there's no change with or without the wrist correction. So with and without the wrist correction. But then there's this massive decrease when you apply the low frequency extension. So for example, I remember catching my mom one night before bed, shaking her arm around a bunch while watching television to try to meet her 10,000 step count goal. But if she wanted the massive step inflation, she could have just applied the low frequency extension and doubled her step estimates, which I thought was kind of funny, but not great scientifically. Now, this was interesting. So I made these heat maps to look at how differences in correlations between algorithms might change with or without the risk correction, with and without this other low frequency extension. So all of these boxes in both graphs are supposed to be pink, and that would indicate no change in correlation. Again, they're supposed to be standardized, and they have been standardized and previously validated. Again, expecting no change in correlation. And there isn't much of a change with the low frequency extension, but with the risk correction, it clearly and it very inconsistently worsened associations. These are massive changes. We shouldn't be seeing any of these colors. And again, this was really surprising, especially for equations that, again, are supposed to give objective measures of physical activity. And this unpredictability is, one, unacceptable, and then two, hadn't been fully recognized in the literature. This was also unexpected. So this equation never classified physical activity as sedentary. So if you sit on the couch all day and watch Netflix, and you use this equation, you would always be reported as being at least moderately active. And then on the flip side here, if you apply this risk correction, you would never be reported as engaging in physical activity, in vigorous physical activity. And I modeled this mathematically. So physical activity is defined as vigorous in this red zone here. And the red and the blue lines represent activity reported without the risk correction and then with the risk correction. So the risk correction essentially transforms this data in a piecewise linear manner so that one will never be reported again as achieving vigorous exercise at these higher extremes, which you would without the risk correction. So when I first saw this, I remember thinking of my cross-country teammates, and if they weren't feeling like a really cold Iowa winter run, they could completely just get away with saying, "Ah, coach, we did run, you just might not be seeing it. It was just the risk correction." And now here, oh, this was really important. So even in patient populations, so these subjects had chronic pain, we saw incredibly high measures of time spent in moderate to vigorous physical activity, which can be an important clinical indicator. So this red bar is the daily recommendation for a healthy adult. And this didn't look right to me. It didn't make sense. So I asked why was this happening? So to kind of try to conceptualize and visualize what's going on with algorithmic data transformations, accelerometer data used to be measured in what are called bouts. And these are custom time intervals, which require a certain level of sustained accelerations, this green dashed line, in order to be recognized. And so 10 minutes in between these yellow bars, 10 minutes is a common bout duration. So let's say that I were to wear an activity tracker during a javelin workout with short breaks here in between different exercises, or say you were to just run up a flight of stairs. So this activity wouldn't be recognized by these bout-based equations because I wasn't continuously active for at least 10 minutes. And so this is the data that might get spit out. And now using bouts is great, and especially used to be great because it didn't require as much storage space. You were compressing things, but storage space isn't a limiting factor anymore. Devices have gotten better, and using bout filters, using bouts and bout-based equations filters out this other activity that might be important. So modern accelerometers have now shifted away from using these processed bouts, and they instead relay this raw data. And shorter duration physical activity is now recognized. And so there's a greater area under the curve, which is an indicate of physical activity, which means that these newer methods should report more physical activity if algorithms, bout-based algorithms, aren't updated to account for this change. So this might explain the large overestimation that we were seeing. And so I proposed a follow-up study to look into this, where participants will follow an activity protocol that I wrote, and we'll use multiple algorithms again and assessments, several of which I developed to see how estimates compare to these national recommendations. So I'm no longer actively involved in the project, but data collection is underway, and I'm really excited to see what happens. So altogether, this emphasizes a need for standardization, as Andre had mentioned, especially in research and medicine. So these equations need to be well understood, and they need to generate reliable measurements. Let's say you're wanting to see if a drug is more or less effective when someone is or isn't physically active. As a researcher, you need to measure physical, and you choose to measure physical activity using accelerometers. You need the measurements to be accurate. So let's say that you're a physician and you prescribe physical activity as part of a stroke rehab program. You can monitor a patient's activity and then personalize treatments accordingly. So the clinical applications, again, are growing exponentially. Christina gave an amazing example of another application in Parkinson's disease. There's many, many more examples, and they will continue to grow. But overall, the big takeaway is that different algorithms are not interchangeable. And the same concept applies to other measures, other types of wearables across the body, other measures, other outcomes, for example, sleep. And the outcomes are dependent on the analysis specifications. So users really need to be aware of this, especially in the healthcare research settings. And more studies are needed to better inform physical activity guidelines. So thank you very much. And Zoltan, I'll turn it back to you.
Thank you very much, Marissa, for this wonderful presentation. And I think I'm very grateful to all three speakers. So Kristalina showed us how to use these devices in monitoring disease. Then Andre was talking about how to use wearable technologies for neuroscience and psychiatry and what are the problems. And not necessarily, he was talking about neuro recordings. I think it was much more general. For instance, from your mobile phone use, you can actually detect depression or maniac phases, or even from your shopping behavior, somebody can tell whether you are pregnant or not. So these are incredible. AI technologies can detect all these. And then finally, Marissa was talking about the algorithms that we use, how can change the analysis. So I think these talks demonstrated actually the key features of NeurotechEU. As you remember, Neurotech was originally proposed in the mission statement that we aim to build trust in new neuroscience technology that can help improve outcomes for health. So that was Neurotech's mission statement. And through this European collaboration, we aim to understand the cultural differences that influence how our citizens engage with the new technologies, and we will share and learn from each other in order to improve outcomes. So I think you agree with me that the wearable technologies, distant monitoring, big data, combining this data with genetics and perhaps with consumer behavior, mental health, this is actually central for NeurotechEU. And that's why I put Nick's email address into the chat function, because we are looking for contributors for future events in digital monitoring and health and disease. And if you would like to contribute or propose a friend or an expert, the more international, the better. And then perhaps a topic. And we also have some venues. As you can see in the background of Kristalina, we have Reuben College, who would be very happy to hold some of these events. So we still have a few minutes for a few questions. So if anybody has some questions in the chat function, then perhaps we could answer these questions. While you are typing it
Marissa already.
Sorry?
I think there's one for Marissa already.
All right.
I see one here from Jonathan Brownlee. So the question is, how did it happen that these evidently unreliable algorithms models became accepted as mainstream, either by industry, medicine, or academia? Can we learn from that failure to avoid that kind of normalization of untrustworthy information? That's a great question. And so to address the first part, I think that one reason some of these algorithms and models became widely accepted was because when they were developed, they looked great. The studies that were used to validate them were performed in controlled laboratory settings. So you might have a participant on a treadmill, you're walking, you're running, you have some oxygen monitors, you have these gold standard, for example, energy expenditure measures, you can count steps right there in the lab, and you were getting this great set of correlations. But then as soon as you turn to lifestyle, physical activity, so not tightly controlled laboratory conditions, I'm running up and down stairs, I'm hitting my hands on a desk, I'm washing windows, you get much more variability. And there's this huge disconnect between the controlled estimates that you're supposed to be seeing on a bike or on a treadmill, and then what we actually do in daily life. And so I think that disconnect, especially at the beginning in some of these preliminary algorithms, generated some of the massive differences that we see. And so transitioning into the second part of your question, and a lot of emphasis has then gone into machine learning algorithms that are based on validated using activities of daily life. These are errors and mistakes, I think, that were made in previous sets of algorithm development that the research community has learned from and continues to learn from. And I think that with advances in machine learning and artificial intelligence, the algorithms will continue to get better. And we already have seen or have made drastic improvements. And again, touching on the last part, will help improve how trustworthy these algorithms and the research can be. I hope that answers the question.
I don't see other questions, but I'm sure we have lots of questions to each other. So I have a question to Andre, actually, that if you think about the impact of these wearable technologies in neuroscience and psychiatry, I think it's right now it's a bit disappointing, no? So what do you think, what is the limiting factor? How could we accelerate this progress?
So I think disappointing to a certain extent, yes, I would agree. In the application specifically to those areas, I think there's been a number of attempts to tap into mental states indirectly, right? From correlates such as heart rate variability, for instance. There's a lot of wearables out there that are looking at that. Sleep monitoring is also huge, and it's being tackled from all kinds of different directions, except from the brain side. So I think what's going on actually, and one reason why the outcomes have been a bit disappointing, is precisely what I said just now. They are being tackled through indirect correlates of something which is the province of the nervous system and the brain. And I think really what hasn't emerged yet, in my opinion, is a brain wearable with particularly good form factor and a high quality output in terms of data quality. So I think that's what's holding us back. It's using the wrong sensing to take on things that are actually, you can only get reliably. I mean, how can you do sleep staging, accurate sleep staging, without
just from the noises you give?
Yeah. You can detect if someone's moving or not. Okay, sure, I don't dispute that at all. But sleep staging, that's a whole different ballpark, right? So once we, I think this is it. It's we need to get the brain measurements. There's no other way around it, and there's progress in that area. So I really think this is an area where we're going to see vast and very fast improvement over the next couple of years.
Thank you, Andre. And I just have a quick question to Kristalina, that in your experience, what is the compliance of the patients? So when you introduce these devices to them and then you explain that you would like to monitor them from home, what are the issues you encounter?
Well, that's a great question, and something we have to address every time, Zoltan, especially when it's something new, and especially for patients who are not really used to wearing, devices like we do, right? The Fitbit or Apple or whatever else is out there. Some patients are quite reserved at attempting to use relationship you built with these patients over the time. And I guess, Andre, you've mentioned that at the beginning. It's not about the science and the output. Is about how you approach what you're asking them to do. And from personal experience, having a study where you follow patients and you become friends with them and their families, understand what goes on behind the scenes, it's crucially important in order for them to start trusting us that what we are proposing actually potentially be extremely helpful in helping them choosing, their husband, their wife who might have Parkinson's to get into clinical trials. So sometimes they are reserved. We have, surprisingly, to my surprise at least, a number of our patients, and especially the people who come forward, are very keen. They might be engineers themselves. I've got software engineers, I've got technologists, I've got all sorts of backgrounds, which a lot of the times is very interesting because they do personalized medicine without even asking them. They will send us, information saying, this is what my tracker shows and this is what your tracker shows. What do you think? They will do the analysis sometimes for us. And I find this interaction very interesting because we really need to think, again, going back to you, Andre, we really need to think outside the box. We're talking about human and personalized medicine here, which one doesn't fit all. And if we don't understand the needs, not only for Parkinson's, but things like dementia, Alzheimer's, psychiatry, neuropsychiatry, which again, we've discussed today, we have to step back a little bit and think, what are we trying to measure? Who are the people behind these disorders, and what's the best way of approaching it? And again, Zoltan, if I could say, and just, with the colleagues here, standardization and talking the same language is very, very important. And I'd love to hear from Marisa and Andre, as we continue to chat about this. I have found that sometimes you're lost in translation if you try to collaborate with different technology companies, different pharmaceutical companies, but also colleagues from bioengineering, for instance. When I first started working with my colleagues here, we were just not speaking the same language. I was saying it's wide, they were saying it's wide, but it was very different shades of wide that could exist. So it's very important that we somehow make sure that we're all on the same frequency so that we can target this. And I wonder if my colleagues here would like to comment on this.
Yeah, I think that's a really important point. And I think it's a challenge in any emerging field such as this, and in particular an emerging field which is taking on clinicians, data scientists, software engineers, neuroscientists, physiologists. And as you say, these are all very different languages to speak. I think increasingly they're getting closer, also drawing examples from other disciplines where the use of wearables has sort of progressed a bit further. But it's still an ongoing issue, I would agree.
We are running a bit late, but I would like to say thanks to all of you for contributing to this introductory event. So we will have many more events, and hopefully this will be part of NeurotechEU and also Rubin College and St. John's College Research Center. So we are looking for contributors for future events, and please contact Nick Irvine if you would like to contribute, propose a participant, topic, or venue. And it can be anything, as Andre just summarized. It can be from ethics to neuroscience to engineering. And but I'm very grateful to all our three speakers, and also I'm very grateful for Nick Irvine for putting together this session. And I'm also very grateful for your attention. Thanks a lot for all of you. Bye.